Thank you very much, Angeles, for the very kind introduction. I feel honored and humbled by having received the BALDI Memorial Award. It means a lot to me, and I'm happy to share with you some insights into the adaptation strategies of plankton in alpine lakes. I was also very happy when I received the news about this award, because somehow my research during the last 30 years connect to that of Edgardo Baldi that you can see here on the left, namely the connection to his research on mountain lakes and on plankton. Here one of his classical studies about the high mountain lakes in the Monte Rosa region. And before I start, I have a very long list of people to thank, even longer than my talk. And as you can see here, these are some photos of the main collaborators, co-workers during the last years. Those with the green point are those affiliated to my university in Innsbruck. And I will not be here giving this talk for this award without the help of all these people and many more I am not able to show here. So something right at the beginning is to express a big thanks to you. Also, I would like to thank the BALDI Memorial Award Committee, and especially Cristiana Calieri, who nominated me for this award, and also the founding agencies that have supported my research during the last 30 years. So looking back at the start of my career in Austria, I would like to acknowledge also what I think are three pioneers of alpine limnological research. The first one, Heinz Löffler, with whom I have little direct contact, but reading his publications in English and German showed me how diverse those ecosystems can be, as he studied lakes, high altitude lakes in the Mount Everest region, Himalaya, but also in Africa, the Bali Mountains, and South America, going from Chile to Peru. The second one, Roland Pechlaner, from the same University of Innsbruck, who I think put the research on alpine lakes in the international arena, and he was part of the International Biological Program in the 70s, and he has many different interests. He's just one representative publication about the factors controlling primary production in those lakes. And finally, Roland Senner, whom also was my PhD mentor, and he again took a very different approach, looking at how different global changes affect particularly the chemistry balance, the water chemistry in those lakes. And here one example of that publication about climate effects and acid deposition in nature. So Roland Senner also made possible to have a modern research station at the shore of Lake Gossenkölle, a kind of iconic lake for us, because we have made more than 150 studies in that system, which is part of LTR and part of GLIONASS networks. And the station and the lab have been really key to reduce the burden on the difficult logistic needed when one studies these ecosystems, particularly when experimental work is involved, like in my case. And I think that is a problem that is still one of the reasons why we know little about those ecosystems, particularly outside Europe and North America. So the interest for studying alpine lakes or high altitude lakes was really at the cradle of limnology, and it started very early, not only with people like Baldi, but also Otto Pester in 1929 published this study about the high mountain lakes from the Alps, and this very well-known paper from Evelyn Hutchinson in Nature in 1933 about the high altitude lakes in Ladakh, India. But as you will see in the next slide, it took quite a while to this knowledge to increase. So as you can see here, even if it is not an exhaustive search for all publications, I think it is clear that there was a very large gap from those very early publications until 1990, about 60 years, where you can see that the number of publications on alpine or high mountain, high altitude, high elevation systems started to decrease more or less in an exponential way, but still, if you look at the number of publications per year, it's a very small fraction of what is the whole output on limnology. So this is somehow contrasting with the knowledge that mountains are the water towers of the world. So they are really crucial for a very large fraction of the whole human population. And also we know that mountains have a high ecosystem, species and genetic diversity, and also have been key to understand evolutionary adaptations. So they are relevant for the ecology we know. And further, mountains, whether low or high, as you can see here in this more or less recent mapping, are found in all continents, and that is really important at the time when you are interested to do comparative ecology or comparative limnology. Also, I would say that the remoteness of most alpine lakes and the low direct human impact on their catchments make of them perhaps the best freshwater ecosystem we have to detect the effects of different global changes, because whenever you see a change in a lake or in a lake district, you can be sure in most cases that it has nothing to do with the impact of human activities in the catchment. So we have to do something to increase even more that knowledge. So before I continue talking about alpine lakes, you may have the question, but what is exactly alpine? Or why do you see many publications where alpine is not used, especially publications coming from Europe, but high mountain or high elevation lakes? And this has something to do with trying to avoid the confusion with lowland lakes which are located in the Alps, and obviously alpine has nothing to do with the mountain change, the Alps. But you may also think, yeah, but what is really high? Yeah. So I don't want here to give an official definition, but mainly to stress the importance they have to do when you cross the tree line. And the position of the tree line actually defines the start of the alpine zone, especially in the temperate region. And this is because this very sometimes very clear ecotone largely changes the strength of connectivity and interaction between lakes and the terrestrial catchment. And as you can see here on the figure on the right, the position of the tree line actually changes with latitude. So it's not a matter of elevation. So we find lakes about tree line in all continents at different elevation. Now this definition of alpine lakes as beyond the tree line is not perfect. And as you can see in the next slide, lakes, for example, in North Chile, located about 4,500 meters in the high altiplano, they don't really fit this definition because there are no forests below the high altiplano. Anyway, those lakes share many characteristics with the alpine lakes. So alpine lakes have mostly glacial origin and share usually many environmental characteristics, some of them which are really challenging for the biota of those ecosystems, like for example very low water temperatures on average during the year, very low concentrations of nutrient and chromophoric or colored dissolved organic matter and DOC, very strong light limitation during the ice cover period, and very high incident and underwater solar UV radiation levels because those waters
are typically very clear, especially when they have no influence of glaciers in the catchment. And in fact, these harsh environmental conditions typical of alpine lakes, and not only of the alpine lakes, this is something shared with also polar lakes, they have driven my research interest to understand how organisms have adapted to them and how they may respond to different global changes. And in particular, the extreme water transparency that I mentioned before, and the contrasting light conditions between ice-free and ice-covered situation was something that caught my attention right at the beginning when I started to work in 1994.
So what I would like to share with you today is first a topic that we started to study in 1994, and this has to do with photoprotection against potential damage of UV radiation in aquatic freshwater organisms, and I will refer mainly to planktonic ones. And what do we know about the drivers that control different photoprotective compounds or strategies? The second topic is something we learned recently on how planktonic microbes harvest solar energy with a dual phototrophic mechanism, a kind of hybrid motor. This is something journalists like to say, and the implications this may have for production studies, something I would like to discuss with you, and I think is very exciting. And on the last topic, which it reads a little bit cryptic, on minimizing mortality, my friend, the enemy of my enemy. This is storytelling of viruses, on new viruses and good viruses. So I will leave it here, and just to keep away your interest. So the interest for the first topic, as I mentioned before, started in 1994 with the accidental discovery of UV absorbing compounds in plankton from that lake I mentioned before in Gossen-Kelose. And at that time I was interested to understand whether the attenuation of UV radiation changed over time, and particularly during the period of the year during the ice-free season where the deep chlorophyll A maximum develops, which is typical of clear aquatic ecosystems. And one very unexpected result was that, as you can see here on the lower graph, there was a very significant increase in the attenuation coefficient in UVA wetlands, and that was much more pronounced than in the UVB, and the change was particularly clear in deep water layers. So these results pointed first that the change could not be explained by changes in DOC or chromophore dissolved organic matter concentration, because it's just the other way it works. And second, that the development of the typical chlorophyll A maximum I mentioned was somehow related to the increase in the attenuation coefficient. Now the question was, what could absorb so strongly in phytoplankton in that range? So it took really a while to find out later in the literature that, for example, corals and their symbiotic dinoflagellates also have UV absorbing compounds. And luckily I met Ferran Garcia-Pichel at that time as Max Planck, that was able to quantify those compounds in marine and terrestrial cyanobacteria. So with him we discovered for the first time that those UV absorbing compounds are the so-called mycosporine-like amino acids. You see here in the middle two of them, which are very dominant in the plankton of that lake and many other lakes, like chinorine and Porphyra 334, and that was the first discovery
of mycosporine-like amino acids for fresh waters. So nowadays we know at least 42 mycosporine-like amino acids that we also like to call natural invisible cytoscreens, and which are widely distributed among not only freshwater organisms but also marine. So that hints to the evolutionary importance of those photoprotective compounds. And as you can see here, they are compounds which are rich in nitrogen, and these are secondary metabolites which are produced mainly by algae. Micro and macroalgae are known to synthesize them, and of course the detection and quantification of those compounds took also a while to standardize, to have at least secondary standards to identify them, and our group invested quite some time on that. So one of the reasons why those mycosporine-like amino acids are widespread is that on one hand, as I mentioned before, algae can synthesize them, but organisms which cannot can accumulate those compounds through the diet, as we know, for example, for zooplankton, for copepods, as you will see later. So one of the first questions we have is how variable are the concentrations that copepods can accumulate on those of those MAs. And the example that you see here is from field studies on the copepod Cyclops avisorum, and each single point is a population from a different lake. And what you can see is a very strong relationship between the concentration of MAs accumulated in those populations and this relationship with what we call the depth refuge. So a parameter which is calculated by the ratio of the UV penetration, in that case 320 nanometers, it doesn't matter, you can take whatever wavelength you want, and the maximum depth. And that is an important proxy for the physical refuge offered by the lake, and that is very important for organisms undergoing vertical migration. And it's very, very strong that that parameter is plain in that region, lakes located across an elevational gradient with 86% explained by that, which also depends, of course, on the different UV stress conditions that the source, the phytoplankton, is exposed. Another important step in that research was to demonstrate how efficient mycosporine-like amino acids are in protecting those copepods, and especially whether MAs are more efficient than other photoprotecting compounds I will talk in a minute, like carotenoids. So in that case, the experiment that by the late Robert Muller with Leptodioptimus minutus was very interesting in that we were able to transform through the diet the concentration of the original population of that copepod, so that we got two different phenotypes. On the left side, a phenotype with carotenoids but without MAs, and on the right side, a phenotype where you see is very clear, no color, but with MAs. And the results of one of the main experiments in that publication is very neat, and in showing that the phenotype on the left side without MAs have a very strong mortality after exposure to UV radiation, and as you can see, a rapid decrease in the survival. And the phenotype, on contrast, on the right side with MAs was not only able to survive, but even in that week was able to slightly grow. So that showed for the first time how efficient MAs are in screening that damaging radiation. So mycosporine-like amino acids are not the only photoprotected compounds found in aquatic organisms, and carotenoids like astaxanthin, which is the most typically found secondary carotenoids in zooplankton, are really pigments, and this is a large family of carotenoids of more than 1,000 different compounds, which are very, many of them are very helpful in reducing the oxidative and photooxidative stress caused by solar radiation. One of the main differences with the mycosporine-like amino acids is, first of all, they lack nitrogen, so it's mainly a carbon, very long carbon molecule, as you can see here. And second, carotenoids like astaxanthin are really pigments, so they have a color, and therefore they don't screen directly like the MAAs in the UV range, but as I say, they are powerful antioxidants. Whether copepods accumulate more of a pigment like astaxanthin or more of invisible mycosporine-like amino acids make a difference when visually foraging planktivore fish is present, because it's clear that they can much easily detect red copepods than those which are translucent. So copepods have a very large phenotypic plasticity, which is partially driven by the presence of predators, especially, as I say, visually oriented predators. And the next example I will show is from Greenland, has nothing to do with alpine lakes, where, of course, fish have been introduced, but they don't belong to the natural food web. And you will see how different fish species affect that balance between these two different kind of protective compounds. So the rationale of this study was that different feeding behavior between Arctic char and stickleback results in contrasting level of those two different photoprotective compounds. So the idea is that stickleback, as you know, are strong planktivorous fish species, and that will cause a reduction in the pigments in the astaxanthin, but not change concentration of MAAs. And that is exactly what you can observe in these two graphs, where astaxanthin levels were not significantly different in lakes with char compared with those without fish. So on the left side, the red cross indicating no fish, but significantly lower in lakes with char and stickleback, so the third treatment, SBC, or just stickleback. Whereas for MAAs, there was no significant difference, and that shows exactly the advantage to attain photoprotection when using MAAs when visual predators are present. Now, if you go to other regions of the world, like for example here in Himalaya, lakes located about 5,000 meters of elevation, where never fish has been introduced and where copepods represent the top of the food web, one will expect that concentration of carotenoids are probably the highest, and in fact I found that were the highest reported until now. But one very striking result was that even if MAAs we know are more efficient in protecting against UV radiation, and those lakes receive much higher incident UV radiation than those in the Alps just because of the elevation difference, concentration of MAAs were undetectable in phytoplankton and in very low concentrations found in copepod, which integrate that signature over much longer period of time. So that's puzzle what we knew about the Alps, the lakes in the Alps, and forced me to think that there are other factors that make a difference. And one of those factors was the possibility that different conditions on nitrogen availability or better say, nitrogen limitation, may affect the balance of what phytoplankton can synthesize and later copepods can bioaccumulate. And the reason is what I explained before. Astaxanthin is mainly a carbon molecule, whether MAAs depends on which ones, but they have relatively high concentrations if you look at the stoichiometry of nitrogen. So the idea about nitrogen came also from knowledge about the process that companies that sell astaxanthin for human consumption use to produce it. And what they do is to stress algal cultures under high solar radiation levels, and at the same time they limit the culture with nitrogen. And what caused this double stress is that the algae switch to a direct carotenogenesis where astaxanthin, which is a secondary carotenoid, is produced. For example, in the case of copepods, the copepods have to transform primary carotenoids like beta carotene into astaxanthin before they can be accumulated. But if they are able to feed directly on a source where astaxanthin has been produced, of course that goes directly into that process. So what I started to look is at different nitrogen hemophilic deposition in different parts of the world, and used the wet dissolved inorganic nitrogen deposition over a certain region as a proxy for nitrogen availability. And what you can see here is a very strong relationship between that parameter and the concentration of MAAs accumulated by different copepod species in different parts of the world, with the highest concentration found in the Alps and the lowest one in the Himalaya. So that's suggested that nitrogen are also responsible for how efficient MAAs can be synthesized. I have no time now to show you experiment I have done in Nepal in one of those lakes where the red field ratio was re-established, and we observed an stimulation in the synthesis of MAAs. This is still progress on a work in progress, sorry. So in summary for this first part, we can say that at the research level or at the synthesis level, exposure to sunlight, which is the main physiological trigger for the synthesis, is of course important, but that nitrogen limitation that we just discussed is controlling how efficient MAAs can be produced, and at the same time when there is a very strong nitrogen limitation, algae can switch to the direct synthesis of astaxanthin. And I think that nitrogen driver, nitrogen control, is very interesting in terms of the nitrogen atmospheric pollution, which have affected very remote places over the years. The situation is changing, for example, here in Europe, but in other places of the world, nitrogen pollution in the atmosphere is still on the increase. At the consumer level, there are several drivers here that we discussed. The physical refuge as a proxy for how stress are vertical migrating organisms like copepods. Also the visually oriented predators in affecting that balance between colored and uncolored photoprotected compounds. And we also know from the work done by Horacio Sagares in Argentina that temperature, of course, as a variable affecting physiological process is also important. In that case, low temperatures favor the accumulation of mycosporin-like amino acids. So all these drivers affect the balance between astaxanthin and MAAs. The second topic I want to share with you is about what at the moment is unclear probably for you, the dual phototrophic systems in planktonic microbes, something that we started to realize very recently. What you can see here is the temporal evolution about the discovery of the different mechanisms photoheterotrophic organisms have to harvest solar energy and produce biomass. So on the bottom of the slide you see in blue, dark blue, this is just the main discovery, like in the case of the oxygenic photosynthesis. The second year have to do with the knowledge about the molecular process behind. The light blue represents the discovery of that mechanism, certain mechanisms in marine systems, and the green one in fresh waters. So the first three mechanisms are dependent on harvesting solar energy by pigments, whether chlorophyllate and bacterial chlorophyllate. And as you see, there was a big time gap, and we started to get to know that beside the oxygenic photosynthesis, there is another kind of anoxygenic photosynthesis, in which that process oxygen is not produced, because water is not used as electron acceptor, but something else like H2S. Now, for a very long period of time, again, we saw that process is mainly found in anaerobic systems under anoxic conditions. But later in 1978 and much later in fresh waters in 1990, we discovered the presence of microorganisms, of bacteria, which are able to have that anoxygenic photosynthesis, but they live in the aerobic part of the water column. And those microorganisms, they are mixotrophic ones. So they are able already to use light to produce energy, but at the same time, they take up dissolved organic matter, dissolved organic carbon for their growth. The fourth mechanism appeared much later, or was found much later in aquatic ecosystems, and is completely different, because proton pumping rhodopsins are very simple from the synthesis perspective, and they work as a gradient pump across the membrane, where the energy is used to produce ATP, and this eventually can sustain biomass. Now, we thought that these two type of mechanisms, the last two, the aerobic and oxygenic photosynthesis and proton pumping rhodopsins, they have been observed in the genome of many different microorganisms. And what I will show you is the first discovery and first demonstration that one single microorganism can use more than one mechanism to harvest solar energy and produce biomass, something that is called dual phototrophy. So the story starts with one study done some years ago, where we really found that aerobic and oxygenic photoheterotrophic bacteria were, in terms of biomass, very significant in alpine lakes and also subalpine lakes. As you can see here, many of them, they have a really large filamentous form, which increase their representation in biomass. During that study, I was lucky enough to isolate one of those aerobic and oxygenic photoheterotrophic bacteria, as Sphingomonas glacialis, and with the collaboration and with the excellent work done at the lab from Michal Kovalchuk in the Czech Republic, especially the work from Karel Kopetka, that was able to run a series of experiments to demonstrate for the first time the advantage and the existence of that dual phototrophy. So as you can see here on the left side, where we compare the respiration rate and the increasing part intensities of a strain where centrorhodopsin is present, so the orange symbols here, but no bacterial chlorophyll, and the control without both systems, and you can clearly see that, especially by increasing light conditions, respiration rates are reduced in that Sphingomonas glacialis. Also, if you look at the middle panel, you can easily see how efficient ATP is produced in the presence of light compared to the dark control, and the same when you look at the growth under the light conditions and the yield, so the abundance can reach at the end of a certain period, is much higher than in the dark control. So this showed for the first time that the use of proton pumping rhodopsins, in that case a xantho rhodopsin, is really efficient and important for the metabolism of that bacterium, and it saves at the end energy and reduce the dissolved organic carbon consumption, which, as you know, is very scarce in this type of oligotrophic ecosystems. Another important difference between the proton pumping rhodopsins and all other photosynthetic pigments, whether the different chlorophyll or bacterial chlorophyll, is that the rhodopsins, the different ones, they have a very differential use of the solar spectrum, with the rhodopsins absorbing, as you can see here, in the range where less overlap occurs with the other pigments. So that again indicates how important, probably during the evolution, has been the inclusion of that proton pumping rhodopsin systems in order to use more efficiently the solar energy. So how dual phototrophy works in situ? This is something that Karel Kapetka will start to investigate this September, but the idea behind, based on the results of the different drivers we found on the synthesis of these two different mechanisms under laboratory conditions, and which I do not have time now to explain in detail, is that during the short winter days when light reaching the lake water column is strongly attenuated by the snow and ice cover, that is the time where the synthesis of the bacterial chlorophyll A containing photosystems is favored. So that is a process which takes much more time, and the low light conditions actually favor the synthesis of bacterial chlorophyll A. In late spring to early summer, when ice starts to melt and the water is still cold, especially at the surface, under still the ice, photosystems probably cannot use the full potential of increasing light intensities because the cyclic electron transport is very slow for that mechanism at this temperature. So that is where we think xantho rhodopsin becomes more advantageous during the year. And then in summer, during the long summer days, probably both systems work potentially equally well. However, considering that the proton pumping rhodopsin has a much simple biosynthesis, probably that is the most cost-effective system used. And then reaching autumn, the warmer the water temperatures and shorter days with lower light influx may favor again bacterial chlorophyll A expression, and that makes sense with our field results, where we found the maximum of the AAP based detection on the bacterial chlorophyll A in autumn. Now you are certainly thinking, well, that is very interesting, but is this just only one bacterium, or how widespread is that process or that possibility? And well, I have no answer to that at the moment. We know of at least, and this is always increasing, but at least 55 microbial genomes where both genes are present. But of course, one has to demonstrate that both are active. And those genomes, they don't pertain only to alpine lakes, they pertain to different kind of ecosystems, even to desert lakes, or has been found also in springs in National Yellowstone Park. So we think that our hypothesis is that this dual phototrophy is very efficient in allowing microorganisms to grow under very different conditions in a continuous way. But of course, this has to be still proved. Based on the knowledge we obtained from Sphingomonas, I started to think further whether perhaps phytoplankton may have this dual phototrophic strategy. So whether they are able to combine oxygenic photosynthesis in a pronto-pumping rhodopsin. I know this sounds very provocative, but I want to show you some results on that. This January, we started to search for the genes responsible for proton-pumping rhodopsins in our data bank from Gaussian-Colosset of sequences, and we actually found to be present among the main phytoplankton groups like dinoflagellates, chrysophytes, cryptophytes, and diatoms, which suggest that phytoplankton may use this mechanism as well. Of course, a proof of concept is needed. Just because the genes are present, it doesn't mean that they are active. But we know now that they are present also for different lakes, not only alpine lakes. In the meantime, appeared two publications last year from marine studies, which usually marine people go faster than freshwater ones. First of all, the paper by Strauss and co-workers showing again that proton-pumping rhodopsins are widely distributed among different marine phytoplankton groups. And the paper by Andrews and co-workers, which I think is very crucial because they show that in polar waters, proton-pumping rhodopsins alone deliver enough energy to produce new algal biomass. This means to sustain primary production under different light conditions. And this is something is important to understand. The proton-pumping rhodopsins are not photoinhibited like the photosystems of the chlorophyll, and it can also work very efficiently with very reduced light levels. So I think it still is a long way to go, but it shows that that mechanism can be used as an alternative or can be complementary to the typical or classical primary production based on oxygenic photosynthesis. So if this is the case, the new biomass production by this mechanism in phytoplankton challenge really our understanding on how solar energy is used in aquatic ecosystems. It challenged also our way to understand primary production, and we will need new methods because we don't have a method to measure it in situ, as there is no oxygen production or inorganic carbon uptake during that process. And as you know, these are the two methods we have to quantify primary production or photosynthetic activity. So I can foresee that in the next years to come, it will bring a strong research effort on this topic, and certainly we will get to know much more about this and what is the contribution in different type of aquatic ecosystems. And I can also foresee that it's not only important in alpine lakes, but also in other kind of lakes. As I say, it can't function in a very different way without photoinhibition as the chlorophyll-based photosynthesis. So that will be a very, very exciting topic. So the last topic has to do with viruses, and something we are working right now, and a storytelling of my friend, the enemy of my enemy. And I thought to bring here results about our research on viruses because, as you probably know, they are not only the most abundant biological entities in whatever ecosystem you look in, they are also an important mortality source of potentially all aquatic organisms. And as I discussed it just before, they transfer genetic information around. And also, we know that they are an important cause for changes in diversity. But we pay a little attention to viruses among, I think, our freshwater scientific community. This is a little bit different from the marine counterpart. And I thought also to talk about viruses because after at least 2 years of corona pandemic, I want to show you that there are also many good viruses out there. So I try to make this story short. So 20 years ago we started to work with viruses, and at that time we found in different alpine lakes giant viruses. Giant viruses, as the name already implies, they are much larger, usually larger than 400 nanometers, so several times larger than the typical phages you may know in aquatic ecosystems. And at that time we didn't have the molecular methods to characterize them. So we have a project to understand the ecology of those giant viruses, but Chris Bellas, that came to my lab as a postdoc, started to observe that there were a new group of viruses, so-called plankton-like viruses, which were very abundant in Gossenküllesee and Lake Gossekülle. And all of them, as you can see here, were related to microeukaryotes, so integrated into the DNA of most common groups, also of phytoplankton, of protists in general. And he also started to observe, very interesting, that they are not only found in Gossenküllesee, they are found in many different kind of freshwater ecosystems and also marine ones, so indicating that they are really important. Now, the ecological importance of this massive viral integration into protist DNA is that many, if not most, of the plankton-like viruses act in preventing host mortality by the giant viruses in a similar way as it is known for biophages, which were also present integrated in the DNA of most of the groups and which are better known. This biophages is a group of very small viruses that kill also the giant ones. So as you can see in this scheme, both plankton-like viruses and biophages can infect independently the host and integrate into the host DNA and then later being activated to stop the production of new giant viral particles, or can eventually co-infect the host with them in order to cause the same effect. And I think, and we think, that this is a fascinating example of host virus armed races and tripartite coevolution. So the interplay among the host, the giant virus, and plankton-like viruses or biophages has also important consequences, as you can imagine, for population and production dynamics, as you will see in the next slide. So in this model by Yao and co-workers, we can clearly see the difference between having just two components, the algae and giant virus, in the upper panel, and adding a biophage or a plankton-like viruses. So it is in the lower panel you can see that the algal abundance is not decimated as it is in the first case in the upper panel, and that the system has a much higher turnover. And that is, I think, important to think that this may influence also secondary production by the DN release from the algae, which are killed by the giant virus. So part of the population of the algae is killed, but they are not completely decimated because the biophage or plankton-like viruses prevent the giant virus to replicate efficiently. And as I mentioned before, I think in this and similar oligotrophic ecosystems where the low abundance of hosts prevent that the whole population is killed at once, a strategy has evolved that results in a very tight control on mortality rates. And with this, I would like to close the last section and to close my talk. So I would like to leave you with this very nice painting, just indicating that mountains are not only very inspiring for science, but also for many people that goes there
for hiking and many other activities. And just to tell that we need to protect those ecosystems in a much more efficient way than until now. If we look at lakes, probably these are the most pristine type of aquatic ecosystems we have in the world, but that situation is rapidly changing and has already changed by introduction of fish in many of those lakes, by the long range atmospheric contamination, for example with nitrogen, and many other substances. So we need to have a much better protection on those ecosystems. And with this, I would like to say muito obrigado, muchas gracias. Thank you very much again for your attention and your patience.